Hi guys, my name is Claudio Pedrick and my project was making a model plant cell membrane for use in future cryopreservation research using molecular dynamics. So specifically, we focused on understanding cryopreservation issues. So one of the issues that we've got that occurs during cryopreservation is ice formation. And ice formation can occur inside and outside the cell, so intracellular and extracellular. Intracellular is always bad because ice kind of keeps growing and ends up penetrating both organelles and the cell membrane, resulting in cell damage. Extracellular ice can do that as well. However, it also results in locking up the ice in those ice crystals, which then results in dehydration of the membrane as water starts to try and equilibrate with the now higher solute concentration outside the cell. To try and prevent ice formation, we introduce cryopreservation agents, or CPAs. This is because they prevent the ice crystal lattice from forming. But they also result in a higher solute concentration and therefore a dehydrating effect on the cell membrane. And as you can see here, we have two different types of CPAs we can introduce, penetrating and non-penetrating. Penetrating, as you can imagine, goes into the cell and therefore counteract that dehydrating effect a little bit. And non-penetrating CPAs do not penetrate and therefore don't counteract the dehydrating effect. Our research specifically focused on modeling plant cell membranes, like I explained, but we wanted to look at the different hydration states with these membranes to see if more water and less water have different effects on the cell membrane, as you would assume it does. We also then modeled them at different sizes, so smaller membranes and then a large membrane. And as you can see here from my visualization, these are the systems that we ran, or some of them at least. Our small full hydrated system and our small dehydrated system have a very big difference in the number of water molecules that they have. We then had another two systems, so one at desiccation with even less water, and one at partial hydration, which is sort of like halfway in between these two. We then had large membranes at those same hydration states. Again, the aim was to see the effect on the cell membrane as you had more lipids, like a lot more lipids. This full hydrated membrane is essentially this one here, like repeated 4 times in a block. And then we simulated those and ran that simulation. And then you can see here what it looks like after the simulation is finished, just a little visualization. The hopes for future is to analyse their APL, so area per lipid, which looks a bit like this. So this is from another research paper. And the area per lipid essentially indicates the stability of the cell membrane as it shows you how much the sort of membrane fluctuates or grows, but specifically it tells you how much the sort of average area that lipids occupy on the cell membrane. The mass density profile and number density profile indicate essentially where molecules have been for most of the simulation. So water, you would assume, would be outside the sort of membrane block. Then you'd have your potassium ions, which are the little red dots floating around, also in that same space. But then your lipids would again be concentrated in the middle. And then as for hydrogen bonding and autoparameters, that is to sort of look a bit more in depth at what's happening at like a molecular level or atomic level, really. And then we would like to again simulate the same systems we've done here, but with different cryopreservation agents. And so in future, hopefully understand why or how these cryopreservation agents affect the membrane, not only when it's at different hydration states, but also when there's like a lot more lipids. So for instance, a small hydrated membrane will have maybe 3 of one type of lipid, but then the larger full hydrated membrane will have maybe 4 times that. So, and depending on how big we can run our simulations, and so the aim would be to see whether or not sort of more lipids results in different effects. So maybe they sort of create these lipid rafts where they end up congregating. And yeah, the software that we ended up using was Blender for the visualization, but for the simulation we used GROMACS. We ran this all on Sotonix. And specifically in Blender, if anyone would like to do something similar with any biological simulations, I recommend Molecular Nodes. It's a module created by a member of the MD community who I think stays in Perth. And yeah, he's done a pretty good job. It's been really helpful at creating a bit more realistic simulations that are sort of nice to explain to everyone what's going on. And yeah, I really enjoyed my time with the Pawsey internship. It's been a great opportunity, and I look forward to working with my friends and colleagues that I've made along the way again. Thank you. Have a wonderful day.